Good morning, afternoon, evening, everybody, wherever you are. Welcome to this already the 18th field day of the Agriculture Genome to Phenome Initiative. My name is Jack Dekkers. I'm a professor at Iowa State University in animal breeding and genetics, and I'm leading the field day efforts and a lot of help from Nicole Scott, who's also online, who's the program specialist for AG2PI. AG2PI is funded by USDA NIFA. We're very appreciative for their funds. And so today we're going to get an international dimension to our field days. We're going to hear about the activities, needs, resources, the opportunities for genome to phenome research in underdeveloped or developing countries. We're going to hear from two centers involved in these efforts, one focusing on livestock and one on crops. And the purpose is for them to highlight the goals of the centers, their needs, and as well as their efforts in building community, which is a big part of AG2PI. So the first presentation is on the Center for Tropical Livestock Genetics and Health, or CTLGH, which is a strategic alliance of ILRI, the International Livestock Research Institute, and the Roslin Institute of the University of Edinburgh and Scotland's Rural College. And we were going to have two speakers from CTLGH, Apollinaire Djekeng and Kelly Watson. Apollinaire had a conflict in terms of change in some of his flights that he couldn't avoid. And so going to hear from Dr. Kelly Watson. And Kelly is a principal investigator at the Roslin Institute. And she is the leader for the poultry efforts of the CTLGH. So with that, turn it over to Kelly. And as Kelly shares her screen, if you have questions, please put them in the chat. I'll address them right away if they're relevant immediately to what the speaker is talking about. Otherwise, we'll have questions at the end, and then after the talk, you can also just speak up and ask your question. So, Kelly, floor is yours.
Thanks, Jack. Well, first of all, thanks very much for inviting me inviting Apollinaire and I to speak today. Apollinaire was very regretful that he wasn't able to make it, but I hope that I can do his slides justice. So as Jack said, I'm going to be talking about the Center for Tropical Livestock Genetics and Health, the CTLGH. I will try and keep the acronyms described because there's lots of them. But yeah, I just want to get on the way and talk about this partnership for livestock genetic improvement in smallholder production systems. So first of all, who we are. So, sorry, let me try and minimize things here so I can see my slides. So we were established in 2015 through some funding from the Bill and Melinda Gates Foundation. And I think it's a nice way to start was to use this quote from Bill Gates in his visit to Edinburgh in 2018. He said, if you care about the poor, you should care about agriculture. And if you care about agriculture, you should care about livestock. And I think that gives a really nice framework for what CTLGH is about. It's a partnership for R&D to support tropical livestock genetic improvement. So the idea is that we carry out the discovery and development and deliver innovations through partners to try and improve the livelihoods and outcomes of people, smallholder farmers in Africa. So the way we set up, as Jack's already outlined, is through three partnering nodes. So we have the Roslin Institute here at Edinburgh University, and in the same building we have SRUC, or the Scottish Rural University College. And our partner in Africa is ILRI, the International Livestock Research Institutes. And together we have a number of partner locations that we collaborate with on a number of different projects. And the overall objective is to meet some of the sustainable development goals. And these are really focused around alleviation of poverty and hunger, gender equity, and response to climate mitigation, essentially. So a little bit of setting the stage. I think it's useful to think about why we are carrying out this research and why we're taking the approach that we're taking. This is a really nice report from the Alliance on Green Revolution in Africa, and it's an African Agriculture Status Report from last year. So today in Africa, 70% of the population depends on agri-food systems for employment, incomes, or nutrition. So it's a huge proportion that are directly reliant on agriculture. So everyone in farming in Africa is a farmer, or at least 70% of them. And between the years 2000 to 2018 in sub-Saharan Africa, agricultural production, so that's crops and livestock, grew at a rate of 4.3% per year. And that's compared to a 2.7% average worldwide. So straight away, you can see that because 70% of the population depends on agriculture and it's rapidly growing, that supporting genetics to improve the outcomes of agricultural systems can have a huge impact. And another way of looking at this is to look at the proportion of the production system that is provided by smallholder or small systems. So this is the share of total livestock derived foods produced from small farms in 2010. And you can see in North America, it's only 8% provided from small farms. And this is in contrast against Africa and Asia, where it's around about 75%. So again, highlighting the fact that if we support these smallholder farmers, we can really transform production in Africa to improve the sustainability of their agricultural systems. And this is some nice data from the ADGG, or the African Dairy Genetic Gains, where in comparing the Kenyan dairy industry to the UK dairy industry. So in the UK, over the last 20-ish years, we've seen a reduction in the total herd size from about 2,500 head down to about 1,800 head in 2020, 2021. At the same time, the average yields have increased. So we were at only less than 6,000 litres per lactation in around about the 1998, up to more than 8,000 litres on the current year. So these are obviously relatively modest yields compared to North America. But then if we look in Kenya, although there's a very small percentage, just 2% of Kenyan dairy farmers are in very commercial intensive systems and can get yields of up to 6,000 litres, the smallholder farmers are producing much more modest yields. So the very best are producing 2,500, and then the average is around about 1,400 litres per lactation. And this accounts for 90% of the dairy farms in Kenya. So straight away, if you consider lifting the average farm up to something similar to the best farm, you can envisage the impact that this would have on the self-sufficiency of dairy production within Kenya. So there's a huge potential to improve, as well as a great need to improve productivity. So I think this highlights the fact that we have this massive range of systems and a massive range of scale and everything in between. So there's a huge opportunity to improve and a huge opportunity to make a massive difference for the systems within Africa. So how do we do this, and what's our strategy? So essentially, CTLGH is centered around the main livestock commodities of dairy cattle and poultry. So we have these two different commodities, and within the work within each of these commodities, we have our farmer-facing collaborators, and these are ADGG, or African Dairy Genetic Gains, and ACGG, African Chicken Genetic Gains, which is now being rebranded almost into TPGS, which is Tropical Poultry Genetic Solutions. Now, we also collaborate with universities and, in some cases, industry. And across these two commodities, we also have the health genetics, so the H part of CTLGH. And this concentrates on really the low-hanging fruit for disease and health challenges in Africa. So the major disease challenges, so things like East Coast fever for the cattle, and parasite resistance, and just general assessment of immune fitness. Now, across sort of bridging the middle area between these three overlapping activities is the reproductive technologies and gene editing. And then wrapping around everything, we have the bioinformatics and the data and computation side of things, and also the biobanking. So as we're preparing for, as we start to improve genotypes, we may displace some of the traditional or local genotypes. And the objective is to preserve those for future generations, and to ensure that we don't lose important genetic variation. So we're also concentrating on biobanking and streamlining that process. So the way that that was the general overview of the way that we're structured. And this is an overview of how we actually deliver to the farmers. So I've already said that we interact with these farmer-facing programs. So in this slide, we have TPGS and ADGG. And these entities are very much about translating for use by the farmers. So interacting with farmers, they're both led by ILRI, and their objective is to translate some of the upstream research that we do and their own upstream research into delivery to farmers. We're very much concentrated on the development side of things, with a sort of continuous sort of reach into the delivery and the application side of things. So we want to be aware of how our research actually feeds down to the farmers, but we don't try and do that ourselves, but we interact with these farmer-facing programs and other farmer-facing national and local programs to make that delivery to the farmers on the ground. So I hope that's given a little overview of who we are and how we work. And now I just want to go through some of the highlights and some of the challenges and opportunities. So first of all, talking about some of the gene editing highlights. And I know this is maybe a little bit peripheral to the ADTPI, but we've made some great progress in this area. So this is some work carried out by Dr. Mike McGrew of the Roslin Institute. And he did this really elegant proof of principle for his work on streamlining gene editing in poultry. So his work is based on manipulation of PGCs, which is the primordial germ cells, which are extracted from the developing poultry embryos. These are edited and propagated in vitro, and then re-injected into surrogate recipient embryos. So they don't produce, these surrogate embryos don't produce their own PGCs, PGCs, but that allows the exogenous edited PGCs to be really successful. So what Mike did is he took the white leghorn, so this is a picture of them, they're white, they carry a dominant white PMEL17 gene, and he cut out this 3 amino acid insertion and allowed all the genetic variation that was present in the birds to come out and segregate. So you can see straight off that morphologically these birds are very similar, but they're able to express other loci associated with feather color that is suppressed by the dominant white. Another example of what he's been doing is trying to look at loci that could be important for poultry heat stress. And he used the poultry, the frizzle feather loci, which is a 69 base pair in-frame deletion in the alpha keratin gene. And the gene location is KRT75. So he carried out this deletion, which was done on a light Sussex background. And this is the heterozygote frizzle feather edited birds. At the moment, we're starting to make these heterozygotes together, and we have some homozygote birds, and they're much more frizzly. And we're going to start exploring the phenotype associated with this deletion, and provides a really elegant proof of principle for the frizzle feather phenotype, with a nice control of full sibs, which will be wild type and heterozygote. Now coming on to some of our cattle editing, and we've looked at the SLIC phenotype here, and it's a truncation of the prolactin receptor, which brings about this very short hair SLIC phenotype, which is naturally segregated in the Senapol genotype. And in response to increase in ambient temperature, you can see that the SLIC is much more resistant to the increase in ambient temperature through the day compared to its wild type, its sibling, which is not SLIC, and the wild type. So our colleague Simon Lillico at the Roslin Institute has developed a methodology based on electroporation, which will be easily translatable into African labs to carry out this edit, or carry out gene editing in cattle embryos. And this was a proof of principle at the Institute here, looking at the SLIC. And these two calves, 007 and 008, were both mosaic calves for the SLIC. And I think you can see that the hair phenotype is quite apparent as a result of the editing. So now moving back into something that's a little bit more core to AG2PI, and thinking about some of the challenges that we face working with Africa in terms of the germplasm that we had available to us. So I'm going to talk a little bit about genomics and population structure here. And a good example, I'm going to use the example of a project that we have, and it's also a really nice example of working with really strongly motivated partners. And our partners here are the Rwanda Agriculture and Animal Development Board, and they are placing a great emphasis on research to try and capitalize on genetics to improve Rwanda's agricultural productivity. And they manage, with a project with CTLGH and Jersey Overseas Aid, supported through their Dairy for Development initiative, we have a project to try and look at developing selection for these Jersey cattle. So Jersey Overseas Aid is actually based in Jersey, which is part of the British Isles, but not part of the United Kingdom. So they have their own overseas aid funding. And they like to support something about Jersey. So this is Jersey cattle in Africa, which do actually quite well compared to our normal dairy cows of Holstein Friesian. So what this project aims to do is establish a blueprint for a dairy profit index, using the Rwanda dairy industry as an example. So how do we balance milk yield, fertility and growth in Africa for the greatest economic gain? So considering a lot of the criteria that we would have in a Northern Hemisphere sort of dairy profit index, but tailoring it to the situation in Rwanda. But we have challenge of the population we're looking at in Rwanda. So I've already said that we have Jersey cattle there, but what we also have is a huge amount of admixture across the main extremes of the cattle genotypes. So this is a admixture and representing the ancestry of the cattle that we've genotyped. And we have genotypes on just over 2,000 Rwandan cattle. And what you can see on this PCA is that in the up here we have the Jerseys, we have the Holstein Friesians covered with this green, and with the sort of burgundy colour we have the local genotypes, the more Indicus genotypes. And there's a whole swathe of admixture between the extremes of the Taurus and the Indicus types, but also between the Jersey type, sort of more Sanger type, and the Taur Indicus mixture. And this sort of fourth ellipse here is probably representing the F1s between these admixed Taur Indicus types and the Jersey genotypes. And this is probably something that we don't have to deal with when we're looking at producing a dairy index in North America or the UK, but it's a real challenge of how to deal with this data when we're thinking about the populations in Africa. So a little bit of a challenge to try and optimize things there. But obviously the genomics provides a huge tool and resource to enable us to overcome these challenges. And it's a strategy that we're also using in the poultry side of our projects. So we think that we've laid really strong foundations with poultry genomics, where we've sequenced over 500 individuals across Nigeria, Ethiopia and Tanzania. And this is the PCA of those populations. With down here, these tend to be dual purpose composite hybrid genotypes of poultry down here, with the Ethiopian, Nigerian and Tanzania genotypes. And the challenge that we face with the poultry work is that we identified more than 10 million novel SNPs, with 50% of those shared across geographies. We had actually had the objective of producing a SNP chip, and we actually designed this high density SNP chip, which we called very snappily the AfriChick SNP. We probably should have come up with an acronym for that instead of that mouthful. But we didn't actually move to production because we were very concerned about ascertainment bias. And at the time we started to look at low pass imputation, and we're now working with GenCove for our genotyping, where we use low pass sequencing with the imputation, and that seems to be working really well for us. And this will be in part because of the great genetic resource we have with the in-depth sequencing we've carried out. So the other challenge is obviously the phenotyping. And I think this is a challenge that faces us all in animal breeding and crop breeding, getting really good quality phenotyping, but it's particularly acute when we think about Africa. Our colleagues in ADGG came up with a really novel way to overcome this. And they partnered with a company called Green Dreams Tech Limited, which had developed this really nice system called iCow. And it's really predicated on the widespread adoption of mobile phones across Africa. So people have mobile phones, not smartphones, but more our, sort of normal SMS type, Nokia type phones. And what iCow's objective was to improve productivity of the dairy industry and just efficiency of cattle production by giving personalized coaching and advice to livestock producers, and based on experts in livestock outreach. And this information was provided by SMS messages to their phones. In response to that information, they actually provide quite a lot of routine information on their cows' health, growth, and productivity. And this is the data that ADGG has captured. So it's a kind of a symbiotic exchange of data and information. Now, the data is really messy, but against the backdrop of really having nothing from these, how these cows are doing, then it's really important and really useful data. So I think that's really nice to think about innovative ways to capture data. And of course, now across the livestock industry, we're seeing this data revolution and this revolution in using sensors to try and capture data. So we have smart satellite collars that are looking at animals' movements, cud chewing habits, and even down to the ambient temperatures and their temperature. 3D cameras that's assessing using facial recognition technology and also recording conformation and estimating body mass. Pedometers that are looking at activity levels and timing when cows are coming into estrus to time appropriate AI visits, and things like tail movement sensors that are trying to determine when cows are actually going into labour to help with calving. So there's this huge wealth of data that could come in, and I think that there's great opportunities for that data to have an impact, in particular in African systems. But I have a word of caution here, because I'm involved in a project in the UK, but where we're looking at ducks. And I'm going to take a little segue here. So we're using sensor data to track the movements of ducks, and we're using a system from POSIX, which is based on ultra-wideband technology. And the objective is to understand components of reproductive behaviour, voluntary activity, and leg health. And we're working with a really capable postdoc, Cami Betts, here at the University of Edinburgh. Now, we have these POSIX systems, these POSIX transceivers on the legs of ducks. And Cami was really excited when the first lot of data came in, except then she looked at it and it was quite alarming how much noise was in the data. So on the right here, on the left here, we have a stationary tag which moves over 300 meters within a 24-hour period. A tag on a duck is apparently moving nearly 6 kilometers, and movement is quite often outside the pen, which we know the ducks aren't moving outside the pen. So it was a bit of a shock, and we started to look at how to clean the data. Now it is possible, and we've been collaborating with people in engineering that have experience of sensors, and using a Kalman filter, we've been able to prune the total distance measured, distance moved for the stationary tag down to about 20 meters, and our moving duck down to less than 800 meters. So we think that we're getting to something a little bit more realistic, and we're just going through the process of fine-tuning that filter. But I think this highlights the need to really validate these sensor technologies before we move them into Africa in particular, because it's very difficult to go out into the field when you're working remotely, or when part of the team are working remotely. So for the poultry system, we're focusing on phenotyping in a different way from the cattle. And because of the difference between a cow versus a chicken, and the fact that as a unit, we think of the flock rather than the individual, then there's real difficulties in collecting individual data when it comes to chickens. We hope in the future we'll be able to work with sensors and automated phenotyping. But for the time being, we've decided to focus our activities, and this is CTLGH in particular, not ACGG or TPGS, but we're focusing on collecting data from field stations. And we work with the ILRI poultry facility in Addis Ababa, which is a really lovely and a beautiful, literally a beautiful facility that was, construction was funded by the Roslin Institute and the BBSRC, and was opened again in 2018, so just after the visit from Bill Gates to Edinburgh University. And the reason that we're working with the ILRI facility is that we can manage the data collection a lot better. We have skilled people on the ground, but also we have not just access to the indoor facility, but they also have outdoor paddocks, where this is aimed to simulate the semi-scavenging conditions that are where birds are generally managed in the field. So I put housed in the field, but if they're in paddocks, then they're not really housed. But we're able to manage the data collection and the, simulate these semi-scavenging situations. So we have growth morphology data that we can collect, some disease response parameters, such as vaccine response, end-of-life dissection, so portioning and just looking to see the internal health, and things like parasite load. The other thing that we have at this facility is we're putting in a weather station for monitoring the environmental conditions, so that we can assess the direct impact of the temporal climate change throughout the seasons, but also the indirect effect. So as increased rainfall might affect the amount of scavenging vegetation, but also the parasite load in the paddocks. So that's the end of me talking about the phenotyping, and I have just 2 more slides where I'm just going to talk very briefly about capacity building and outreach. So an area that everybody can get involved in is through the African Animal Breeding Network, or AVNET. And this was, Apollinaire helped establish this. We have partners across Africa, and it's really an objective to transform livestock production and genetics and breeding through supporting people on the ground in Africa. And we're offering, the AVNET is offering training courses, and really starting to try and build a network of animal breeders that can be self-reliant and improve the opportunities and the reach of genetics into Africa and from on the ground. So please feel free to visit the website. I don't know if the links will show up in the presentation, but yeah, visit and you can get involved. In particular, involvement for people that can offer training and support directly for people within Africa is really welcome. The other area where we've actually had some activity is trying to inspire the next generation. So it was a really nice little project where collaborators in the Institute here, the Roslin Institute, through CTLGH, produced this comic book called More Milk Zuri. And the objective is to tell children about what genetics does, and why it's important, and how it can, how the power of genetics can actually be helpful in our day-to-day lives. So feel free to come and download it. I believe it's been translated into a number of languages. But I'll move on to my final slide, and it's the slide acknowledging all our funders and saying a big thank you to all of you for listening and giving our contact details. So thanks very much. And I don't know if I need to stop sharing.
No, you can. Well, yeah, why don't you stop sharing? Thanks, Kelly, very much for a very fascinating presentation. Great to hear about all the work that's ongoing, and the challenges and the opportunities, and also, you know, how the community building is an important part of what you're doing. So if you have questions, please put them in the chat or just speak up. I'll start. So the CTLGH focuses on dairy and poultry, and I was wondering why small ruminants like goats and sheep were not included in that, because I mean, I see them as having a large potential in developing world also.
So that really comes from the funders. So the Gates Foundation are very much focused on dairy cattle and poultry. And so, you know, in terms of the funding, that's where the funding is. As part of the sort of team here at CTLGH, we have a number of people that are looking at small ruminants, dairy goats and sheep. So that is something that we are doing at CTLGH, but it's not a focus, and it's not a funded focus through the Gates Foundation. So really we need other sources of money to work on those important small ruminants, and of course pigs as well. Yeah.
Okay, any questions from people online? Otherwise, I'll have another question. So it was interesting to see the data you showed on the ducks with the sensors, and it reminded me of one of the first field days we had was from the University of Arizona, I believe, was for crops, where they you know, have this big thing going across the field with sensors on it, and even there, they had to. I mean, I was thinking, you know, if you have a plan doesn't stays in place, right? But they had to do a lot of stitching together because of the noise. Is that so? Do you know where the noise comes from? What's the source of that noise?
So I think that the way that the system works is it has these 4 anchors around the edge of the pen. And I think that what we're seeing is that some of the signals from individual anchors is being periodically dropped, and it causes the center of the signal to jump. And that's why when you see the stationary tag, the sort of signal jumps towards the anchors, essentially. So it's really noisy data in that sense. But now that we've worked out how to clean it, there's a huge opportunity there. The other thing that I didn't talk about is that from that data, we also have accelerometry data, and that doesn't drop out, that is complete. And that's what we're using to sort of tune the positional data. So we'll be able to get estimates of the total distance traveled, but not where they're traveling or how they're interacting with each other from the accelerometry data. But I guess naively, we thought that the data would be a lot cleaner. Now, the people that provide the data, POSIX, they're able to turn filters on at source, but of course then we can't validate what we're doing so easily, because we don't know what they're doing to the data at source. So we chose to go down the challenging route of trying to clean it ourselves, but I think it will be more rewarding. And I think as we're in the UK, we're seeing a lot of calls about how to develop sensor technology to improve our phenotyping. So I think it's important as animal breeders that we understand how messy this data can be, and the steps that we undertake to try and improve it.
Yeah, and it's relevant, in the developing world and in the developed world also, for rangeland systems and so on. There's a question in the chat from Dalu Guan. I'm wondering how the African chicken SNP chip works.
So the African chicken SNP chip, we designed it, but we never manufactured it. So it could have been designed for the Illumina platform. It could have been designed for the sort of manufactured on the Illumina or on the Affymetrix platform or anything else that was available. But what we tried to do was to position SNPs throughout the whole genome and not select SNPs based on their minor allele frequency within any sort of breed, but try to make it as random as possible. But that it's always going to be subject to ascertainment bias when we think about it translating into other unknown populations. And this is why we went for the low pass imputation approach. The accuracy of the imputation was extremely high, around, well, greater than 99% when you filtered out low in silico poorer calls. And you get the data, all the data that is there within the population. Of course, that can only be done if you have the imputation data set to run the imputation efficiently. And we had that as part of our phase 1 of CTOJH investment on poultry. Mm-hmm. So it's working really nicely for us. So we won't be manufacturing the chip, but we're able to share the imputations set with requests. So we can do that through GenCove, and they're very happy to do that.
Okay, good. So is it feasible from an economic perspective, the cost?
Yeah.
Okay.
So for us to manufacture a chip and run a chip, we could only get to a fairly modest density, whereas we're able to do that with low pass imputation, and quite economically viable.
Okay, good. A question from Sergi Edmi. How do you foresee implementing breeding within this network? And I'm assuming it refers to the AAB network, bringing in new genetics or something else?
So I think that the situation across in dairy versus poultry is a little bit different. The Gates Foundation actually supports poultry developed poultry coming into Africa, and ways to disseminate it and support it so you don't have high mortality. But there, I think that we want to support local breeding programs that could act as really high quality terminal sires or as a breed in themselves. So that's the strategy that we're taking with the poultry. On the dairy side of things, there's always going to be the desire of local producers to have exotic genotypes to produce within country. Mm. And then it's about getting breeding values that are more appropriate to the performance in Africa. So this is where we can use this great admixture between the Taurus types and the Indicus types to try and understand which of these Western bulls are performing well in Africa, and whether there's appropriate exotic germplasm to bring in over and above others. So the Jersey being an example. But yeah, I think it's about balancing what you might ideally want to do. For me, breeding within country versus what people want on the ground. And they generally see the exotic types as being more desirable to have, even though they're very difficult to manage. And I think that we can just help them being less difficult to manage.
Yeah. Okay, good. There is one more question in the chat, but I think for time we have to move on. So maybe Kelly, if you can look at that and then maybe answer in the chat, appreciate it. Thank you. And so next we're going to hear from Dr. Sarah Hearn. Sarah is a crop geneticist with CIMMYT, the International Maize and Wheat Improvement Center in Mexico. And she is going to talk to us about that center and its what it is involved with, and the challenges and the needs and opportunities. So Sarah, floor is yours.
Thank you very much, Jack. Thank you, everybody, for your attention. Okay, now you should be able to see the right screen.
Yeah.
So, okay, so I'm going to talk to you a little bit about CIMMYT and the 1CGIR. I'll explain to you what that is. Some of the opportunities that we see in the crop research space, and I'm going to focus in particular on maize. That's the main commodity that I work on. So what is CIMMYT? CIMMYT is the International Maize and Wheat Improvement Center. CIMMYT is the Spanish acronym. The mission of CIMMYT is really to focus on maize and wheat science for improved livelihoods. We recently expanded that crop remit somewhat. We now cover other species like sorghum, millet, and groundnut. So the vision is to try to deliver science and technology to really focus on a world with healthier and more prosperous people, really to try to mitigate some of the challenges we have of global food crises, and to develop agri-food systems that are more resilient to climatic factors, more resilient to different policy shocks. Overall, CIMMYT in its history has had annual benefits of about $3.5 to $4 billion, so quite a high return on investment. Around half of the maize and wheat grown in the developing world is based on CIMMYT varieties. In particular in wheat, many of the varieties are directly produced by CIMMYT. And when we look at the capacity development work that we've conducted over the years, we've trained over 10,000 agricultural scientists and experts, ranging from people working in biometrics and genomics, all the way through to extension agents. CIMMYT has around 1,200 staff from over 50 countries. We're quite a diverse bunch. And this image simply shows you the offices that we have in various countries. We're headquartered here in Mexico, but most of our work goes on outside the country. And you can see in the little white dots, they're the offices that we have. And then in green, that indicates some of the countries where we have active projects at the moment. Now I'm going to talk to you mostly about breeding and varieties. But we have a pretty integrated agenda of research. We have two of the main global gene banks here for maize and wheat. So we conserve that diversity and make it available for use for research and development. We develop and improve varieties. We have a lot of work on farming systems. So a variety by itself is all well and good, but that has to fit into the local context in terms of agronomic management practices, mechanization, participatory research around which varieties are the most appropriate in a certain context. And then really we focus on increasing impact through integration with social sciences, big data, focusing on gender and youth, what are the particular needs within target geographies, and then looking forwards with foresight and impact assessment work to try and get ahead of the curve a little bit and predict what is going to be needed. And as I mentioned earlier, across all of these, we have a lot of activities on capacity development and making sure we work in effective partnerships with local agents to deliver desired technologies. So what is the CGIAR? It isn't a cigar. We don't work on tobacco. It's an old acronym. This stood for the Consultative Group of International Agricultural Research Centers. It was formed in 1971. Both IRRI, the International Rice Research Institute, and CIMMYT were founding members. And it's a global partnership of 15 different centers, which work to advance the agenda of agricultural development. Now, as some of you may have heard, we've been going through a big reform recently. This is called 1CGIR. We are meant to be getting a new name. We haven't landed on that yet. And all but 3 of the centres, CIFOR, IGRISAT and World Agroforestry, are not currently joining 1CG. All of the others are. And the aim here is really to try and integrate some of the capacities, capabilities and knowledge that we have. So the old acronym of the old statement of you can move further when you move together, that's what we're trying to do and really start to tackle in a more joined up manner those sustainable development goals. So you've got the SDGs, really we're focusing mostly on poverty, hunger and climate adaptation. We work through a series of impact areas. So we've defined nutrition, health, food security, poverty reduction, looking at livelihoods and jobs, gender equality, youth and inclusion, really making sure that a lot of these technologies are fully accessible to a range of people, and we make sure we listen to some of the needs of gender and youth in the work that we conduct. Climate adaptation and mitigation, we're going to focus on that as we move forwards. And then really looking at environmental health and biodiversity. Agriculture doesn't stand by itself in the environmental world. So as part of this reframing of 1CG, we've got a new framework. So we have 3 main science areas. The system transformation space really looks at agroecology, greenhouse gas emissions from source all the way through to sinks, climate change resilience at a very broad scale, foresight and targeting, policies, markets, value chains. The Resilient Agri-Food System Initiatives, or RAFs, focuses on agronomy, plant health, kind of disease epidemics, modeling all of that. Sustainable intensification, climate mitigation and adaptation through a lot of changes in agronomic practices. And there's some work now being brought in on urban and peri-urban agriculture. And then the area I'm going to focus more on is genetic innovations. This is the part that really is more relevant to the work that we're talking about here and this meeting. And this really looks at germplasm. So right the way from the conservation of germplasm through the use and the delivery of appropriate genetics to the right markets in response to farmer, processor and consumer needs. And then across all of this, we have some regionally integrated initiatives. So it's a little bit of a matrix structure trying to cover all bases. So why are we doing this? Well, this is some information from the 2020 SOFI report. 690 million people, nearly 9% of the global population are hungry. This has actually got worse in the last year, partly in response to the COVID crisis. So this number has gone up by about another 10 million people. And when we talk about this global hungry, most of the global hungry are prevalent in South Asia and Southeast Asia. But when we look at those extremes, there are far more people suffering from severe food insecurity in Africa as a proportion of the population. Now, this challenge of people being hungry isn't going away. The global population is increasing. And at the same time, when we consider certainly cereal production, I've used maize and wheat here as an example, but we can scale this out to most commodities. We have challenges of climate change, varying climate, challenges of new diseases and disease epidemics spreading, and then challenges for access to water, access to fertilizer and nutrients, access to energy as an input when we're dealing with mechanized agriculture. So they're all pushing the trend down in terms of productivity. Through things like agronomy and breeding, we hope to be able to respond to this. But if we look at that gap, the projected demand, that's there in the green dotted line, for both maize and wheat, this was projected by the FAO. And you look at the extrapolation of current trends, they're the lines, unfortunately, which are below, we're going to have a bit of a yield gap, a bit of a production gap. And as I mentioned, this isn't only the case for wheat and maize. We see this when we look at roots, tubers, bananas, and legume crops too. So we have a bit of a challenge. Now, this challenge knocks on in terms of challenges for food prices. These are some recent data from the FAO, the World Economic Forum, did a synthesis of this. And you see increasing food prices over the years, certainly increasing this year in response to some of the challenges. And that cuts across commodities. So increasing costs of vegetable oils, cereals, dairy products, sugar, etc. Now, as I mentioned this year, we've seen a bit of a price hike recently. March was the highest cost of food that we've seen since records began in terms of proportions from the baseline. It's gone down a little bit in April, but still the cost of food is very, very high. And this often disproportionately affects those in less developed economies. If we look back, this is a bit of a challenge, not only just for the affordability of food, but a lot of political shocks that come out from the event of this. We have the food crisis that occurred in 2007, 2008. Wheat and rice prices nearly doubled in this time, largely in response to oil costs, biofuel demands, some export bans on products, panic buying, droughts in major wheat producing areas, and poor harvests. In this period here, we had droughts in major producing countries and demands for biofuel. Now, yeah, we know all about the challenge of the Ukraine crisis at the moment. Energy costs are going up, that impacts mechanization, it impacts things like fertilizer application, and we have anticipations of poor harvests in the Ukraine, which is a major wheat producing zone globally. So again, this is impacting the poor the most, but most countries are now starting to feel the squeeze in terms of food prices. So before I go on to breeding, I just want to highlight a couple of cases where we are using genomics, we are using phenotyping, not directly in the breeding process, but we're using it either to inform that process or we're using it downstream more towards an agronomy-related application. So here, this is an example of an early warning advisory system that we have for wheat rusts. So it includes information that comes from climate models, forecasting, that is all conducted by the Met Office in the UK. We use information from on the ground, so surveillance information. Now this takes a whole myriad of sources. It can come from crowdsourced information where farmers are actively taking photographs and images of what they think is rust within their fields. Surveys where we're actively going out and partners are going out and collecting directly information. And then also, as I mentioned, this genomics application where we're using MinION sequencing to try and get close to real-time diagnostics in the field. Now, all of this information is combined together and consolidated within an early warning unit. And then from that information, we're able to put out advisories and alerts to colleagues and national systems working on the ground to help inform farmers and the national systems themselves of potential challenges to control diseases in key areas. Now, this system has been rolled out now in Ethiopia, Nepal, and Bangladesh. We're reaching close to 1 million farmers. And if we just look back in the last season, Ethiopia from 2021 into 22 had an extremely high risk for a yellow rust epidemic. In addition to this information, we've got data that helps us understand what varieties are grown in which regions of Ethiopia. We've conducted that work in collaboration with the National Statistics Agency, and we've, on the back of that, genotyped a lot of farmer materials. So we've got a very good footprint of what varieties are there. Knowing what varieties are there, we know what levels of yellow rust and stem rust tolerance there are within that varietal profile. So we're able to map out and identify high-risk areas. And then farmers are able, with the support of the extension agencies, to go out and spray to try and mitigate disease epidemics. So it's quite a cool combination of tools, which is underpinned by genomics with this MinION sequencing. Another application is ear photometry. So what we've developed, our colleagues have developed, is a system where we can take images of ears. This can be a standard phone image taken by a farmer of ears that are harvested from on-farm trials. On station trials, yield is pretty easy. We go out and we harvest with combines or we harvest by hand and we get a good measure of yield. In these on-farm testing networks, that is much more challenging. And the reliability of data that is reported by farmers varies quite a lot. So using this photometry system and the ear analyzer software that's been developed, we're able to measure yield parameters based on the images. So average cob size, the number of cobs, the uniformity of that material, the kernel numbers and the size of the grain of the kernels themselves. And taking this information together, we get a pretty good correlation between measured grain weight when we've gone out and sampled and the estimated grain rate. And in addition, when we take these images, we can look at the prevalence of some diseases, some ear rots, and the number of trials with ear rot based on the image detection is much more accurate than that reported by farmers again. So this is another cool application that we can use in this space. Right, now I'm going to talk to you about genetics. So within the 1CG, we have the Genetic Innovation Initiative System, or the Genetic Innovation Science Pillar. And within that, we have a series of initiatives. The first is accelerated breeding. This really focuses, as the name says, on breeding. So from upstream to downstream breeding applications. Breeding resources is an initiative which really underpins and supports a lot of those breeding activities. So it's the use of shared services, whether that's bioinformatics, biometrics, data management, some shared trialing networks, etc. Market intelligence helps inform what the breeding teams should be working on. So really looking at what are the desired traits, what are the important markets, etc. SeedEqual is the system which then takes that improved genetics that have been developed by accelerated breeding and starts to move that out through the seed systems, through towards varietal registration and dissemination to farmers. Precision genetics is an initiative that is just being approved and evaluated at the moment. This really looks at the use of gene editing and transgenic technologies within the space. And at the center of all of this are the gene banks, which we'll talk about in a little bit. So if I look at breeding, this is a conceptualization of some of the CG, NAS, National System, and small medium enterprise breeding networks. So right at the very top end of the scale, you have the germplasm banks. This is where all of that native variation either has come from originally or is still being derived. As you move away from the germplasm banks, which are really the predominant work there is conservation and documentation of that material. Once we start doing any exploration for traits, that moves within the accelerated breeding function. And we have work there that really looks at trait mining and the development of new donors and introgression of important traits. That material then flows into CG and NAS population improvement systems, where we're really focusing on closed systems, highly elite by elite crosses, trying to really drive that genetic gain and accelerate the rate of genetic gain. From the population improvement, we move into early stage testing, which is a collaborative space. The later stage testing moves then more out of the breeding sphere into on-farm testing, seed systems work, etc. And down at the bottom is the indication of how we target some of the particular traits through integration with a lot of the market intelligence information, conceptualization of what is actually feasible from the genetics perspective in terms of available variants, and then using that to further on inform that population improvement space. So looking at some of the challenges that we face, if we look at heat and drought, and I give you an example here on maize in Africa, this was a study conducted across 20,000 yield trials using historical data. And what we see here is for every degree day or day degree above 30 degrees C, we get a lower yield. Okay, so now that's bad enough under optimal conditions. Under drought-prone conditions, this effect is exacerbated. Now, many of the current drought donors and the key parental lines that we use for drought are susceptible to drought stress at elevated temperatures. So really the combination of these challenges of heat and drought coming in a combined manner are really starting to make us squeak in terms of the available genetic variants. Now the CG gene banks, we have 11 gene banks within the CG system. They're the largest and most frequently accessed network of germplasm banks globally. Together they hold just over 750,000 accessions of crops, whether they're clonal crops, seed-based crops, etc. And that's around 10% of the entire ex situ collection held globally, not accounting there for duplications. 94% of all of the germplasm distributed globally in alignment with the International Treaty on Plant Genetic Resources for Food and Agriculture is distributed by the CGIAR, and the majority of that comes from CIMMYT, from the maize and wheat collections. So we have this germplasm resource. This germplasm resource is a great basis of native traits, but as anybody is a breeder in the audience, most breeders will tell you the last thing that they want to do is go all the way back to these land races to use directly in a breeding process. These things are wild, woolly, they're not particularly easy to use. And if we think of that conceptualization of a core breeding unit, where you've got closed populations with elite by elite crosses, throwing a land race in there is not the best of ideas. So this is where we come on to a lot of that trait discovery and pre-breeding work. So we want to make these thousands of years of evolution. In the case of maize, as you see here, this is 10,000 years of evolution, and really ask, how do we make that work for us? Now, at the end of last year, we started a new endeavor. This part is funded by the Bill and Melinda Gates Foundation and maps to the aim for climate initiative. It's detailed as one of the sprints. So the project is led by CIMMYT. It covers a multitude of commodities though. We have maize, we have cassava, which is focused on by CIAT and IITA. Cowpea, worked on by IITA in UC Riverside. Rice with IRRI in the Philippines, and sorghum, which is led by Colorado State University in collaboration with CIMMYT. We also have partnerships with Cornell University and UC Davis, who are working on some of the more back-end analytics and things that really cut across these commodities in terms of the analysis. So what are we doing? We're trying to extract information from the germplasm banks and try to develop new donors for germplasm improvement on the cheap. So we've been extending a concept known as environmental GWAS. So most of you know standard GWAS. You take genotypic data, you have phenotype data of interest, and through combined analysis, you identify QTLs and candidates of potential interest in terms of response to a phenotype. What we do here is simply switch out that response variable, and instead of using phenotypic data, we use environmental data. Now, this environmental data is derived from the collection site origins of a lot of these land races and gene bank accessions. So just like you and I, any germplasm bank collection has a passport. And that passport details in many cases not only the country that an accession has come from, but it will detail the actual collection site. And we are able from that positional data to pull out long-term climate variables that we then use in these analyses. So this was piloted under an earlier initiative, the Seeds of Discovery project. And this was work in collaboration with Ed Buckler's lab, in particular a PhD student we had with Ed, Alberto Romero. So this pilot work was conducted in a GWAS panel. It was a real GWAS panel. This material had been phenotyped. In total, we had about 3,500 landrace accessions represented here. 2,700 of those had good collection site geolocation data. So here you see in an initial analysis where we've looked here at precipitation during the growing season and used that as a response variable. And here you see just one little Manhattan plot. This is a cutout of chromosome 9. And you see an absolutely beautiful peak. We all get excited when we see peaks like this. And this peak really sits on top of the heat shock transcription factor, which has known response and known effects in response to drought stress. So we get signal that is biologically relevant. We tested this further, again within the same panel. Again, remember, I mentioned this had also been phenotyped. And we start to look for target traits. So this is work conducted in collaboration with Jeff Rosavara and Dan Runcie. This particular work was conducted by Dan Gates when he was still in that lab. And so in plot A that you see here, we've got the typical Manhattan plots in response to temperature, precipitation, isothermality, so that's looking at day-night extremes in temperature, and the frequency of wet days. In plot B, you see the genotype by environment effects from the top 1,000 environmental GWAS peaks. So we took a slice of the top 1,000 peaks versus a random selection of peaks. And the G by E selected markers, or those selected from the peaks, you see improved fitness, so improved yield in those environments where you have an environment more similar to the origin where these SNPs have been detected. What does this mean in terms of production? In the main graph to the left, you can see the yield under drought and well-watered conditions. And you see that the SNPs selected under low precipitation environments, or the SNPs favored under low precipitation environments from the environmental data, those entries tend to have a higher yield under drought stress. So we find biologically relevant SNPs that are relevant for particular stresses of interest through these analyses. We can take this analysis further. So this is one of the regions that was identified of interest here. This was under precipitation, looking at precipitation over the 6-month growing period. Here you see this particular peak sits right on top of a gene, which under the provisional gene model, we think is an oligopeptide transporter. In this case, a glutathione transporter. And there's been quite a bit of buzz in the last years about glutathione transporters and their role in drought tolerance. So, okay, that's already giving us a little bit of confidence that this thing is biologically relevant. When we look back into the yield data and we look at those entries and their genotypes, we see that those entries that have the genotype favored in the low precipitation environments have a higher yield under drought. Interestingly, the heterozygote here is also faring pretty well. And in some separate work that we've been doing, a lot of these novel alleles that we're looking at seem to have quite a lot of directional dominance, which is pretty interesting and useful for the breeding work. And then finally, we looked at the expression within this particular gene in completely unrelated expression data under both leaf and reproductive tissue, in this case, tassel in the graph. And this experiment was quite favorably run in well-watered and drought conditions. And you see differential regulation of this gene. So a lot of evidence here that we're not only able to identify favorable or potentially favorable variants, they have biological effect of interest to us from the breeding perspective. And by taking panels of these QTLs and comparing with genotypes from the elite germplasm, we're able to narrow down to those variants that are really novel to the elite germplasm pools. The last thing anybody wants to do from the breeding perspective is recreate the wheel. Okay, now we're scaling this now from, this was done in 2,700 landraces to 17,500 landraces. We've genotyped the entire maize germplasm collection in the International Bank here. And so we're now able to use that information in combination with this collection site data to en masse start to screen for these potentially useful variants. Right, you find QTLs. So what? Now we've got to do something with them. When we look at the breeding perspective, there is a need for new variation in elite-based donors. As I said earlier, the last thing you want to do is go throwing a landrace into these closed populations. So we need to do some improvement or some deployment work, pre-breeding work to try and clean the genetics up so that we can deliver novel genetics in an improved background. So for the major genes and those oligogenic traits with large QTLs, this is relatively simply achieved through a forward breeding approach using markers. On the back of this, gene editing has a huge potential here for validation so that we can actually validate these effects in more elite backgrounds in a much faster way and target and narrow the targeting of the QTLs that we're going to follow forward through. And then when we look to quantitative variation, again, as we all know, this is much more complex, but it is tractable. And I just give you a quick example of some earlier work that we've conducted. This is breeding data that comes from my colleague, Terry Molnar. And if you focus on the yields, which are highlighted with the pink boxes, the germplasm here, those bottom entries that are colored in white or aren't highlighted, these are experimental donors that we've been developing. These have all been developed with a focus on drought tolerance, and they are all in the background of that CML 376, which has been included here as the genetic check. Above that, you can see two entries, which are commercial, are parents used in commercial hybrids currently, which have been used as a check. And all of this data is being generated over the numbers and seasons and locations indicated at the top using 3 different testers. So we've used both the single cross and then inbred line testers in this analysis. And as you can see from the yield data, under drought stress, these entries are outperforming both the recurrent parents and some modern checks. And equally, under optimal conditions, we don't have a yield penalty. So this indicates that we are able to extract value and significant value from these exotic land races as we move that through into the development of new donors. Now, what else can we do with this data? Genomic prediction. So genomic selection is kind of the flavor of the month at the moment with animal and plant breeding for very good reasons. And genomic selection, we know, and we've got some evidence now for that it works well in complex traits, even in land races. Certainly in maize, we've got some very good examples of that now. So there is the opportunity to use genomic selection in the pre-breeding process, as long as we have obviously that good linkage between a training panel and the actual material we're trying to develop. Now, what also we can do is turn prediction on its head a little bit to try and inform breeding. So the two graphs here is an analysis using prediction where we have an observed value. In this case, we're looking at drought occurrence within the growing season. And we've got that observed value at different landrace collection sites. Now, among the collections, these 17,500 materials, we've used part of the data set to develop a training model. And then obviously with the standard cross-validation, we've tried to predict the value of unknown land races, or masked land races, and predict the value of drought occurrence in their collection sites. And as you see, the correlation is extremely good. We're getting a prediction accuracy on average at about 0.8, which if you were talking about a prediction accuracy of that in yield, most breeders would be kissing you. So what I mean about turning this on its head, it's taking this same data, and this is a couple of slides, or a slide that came from Jeff Rossabara, some analysis we did, where we take that same climate data, you take the same genotypes, again, there's an association here through that geolocation, and you develop a model. But this time, instead of predicting the performance of materials, what we try and do is predict for a particular environment of interest what may be the ideal genotype. And we all remember the idiotype breeding, so I'm going to call this an idiogenotype. So it's an in silico, synthetic, made-up genotype. And we think from the data, this may be the best material for this particular location. Now, falling back on the trial data that we had from Seeds of Discovery, we're able to test how well this genotype performs. And here in the graph, you see the distance, so the genetic distance of trial entries from the ideal genotype. And so as you move forward to the start of the x-axis, this is very close to being very distant. And here we have the Z-score for the grain weight achieved in those trials. And you see that the closer the material is to that idiogenotype, the better the performance is in this particular environment. And this has a real opportunity, and we're testing now how well this data transfers then into the elite germplasm, because essentially the large collection that we have in the germplasm bank is kind of the ultimate training panel in terms of representation of genetic diversity. So I will wrap up by thanking a lot of the funders. This is just a smattering of some of the funders who resource the work at CIMMYT. And then, yep, happy to take any questions. Thank you very much.
Okay, thank you very much, Sarah. Very impressive the amount of work that has been done and with the data available and the challenges. Oops, apologies. The challenges associated with collecting phenotypes and but impressive what has been accomplished. Some questions. Let's see. Question from Kelly, our first speaker. She's wondering how the Nagoya protocol has impacted your work on testing and developing new germplasm.
Okay, so I mean, all of this is surrounded by and framed within the international treaties that we have to adhere to. So there's the International Treaty for Plant Genetic Resources, which was mentioned. And then in addition, there's the Convention on Biological Diversity. So we're really working here with germplasm that is fully listed under the SMTA, that we can share under the SMTA. So that has the benefit sharing back to various communities embedded within the framework. And we're also working to update data access and data sharing policies and procedures so that they align more with the SMTA. So as long as we are working with germplasm that's registered under the treaty, we have freedom to operate in this space in terms of the testing and the development of new germplasm.
Okay, thank you. So a related question from Pat Schnabel, and it's related actually to the war in Ukraine. Was reported that the gene bank in Kharkiv has been destroyed.
Yeah.
The question is whether all 160,000, I'm assuming that's wheat, all 160,000 accessions, were they lost?
I had the same question. I was looking at some videos of that at 4 a.m. this morning when I was getting up. It's very depressing. That 160,000 accessions was multiple species. Now, obviously, in Ukraine, wheat is probably the most relevant material within that collection. I need to check with colleagues, and that's on my list of things to do today, to see which of those materials we have backed up within the international collections. So here at CIMMYT, we have safety deposits of material from various germplasm banks. When the collection was lost, I want to use that word, in Syria with Akada, we had the full collection black boxed here. So we were able to regenerate those materials and rematriate them back out to the new Akada bank. So with the Ukrainian collection, I don't know. I hope things are backed up. I mean, Svalbard is a full black box, but yeah, that's on to some follow-up work for me to try and do today.
Mm-hmm. Yeah. Actually, Vivian Bernot just copied a message that was shared among GeneBank staff that relates to this. So
okay,
that's in the chat. So thanks for that, Vivian. Question from Sergey Edmey. On a side note, is the decision out that there is no value in introgressing tropical with temperate maize? No,
there's a lot of value there. So we have, and temperate into tropical both ways. So here we have some active programs looking at materials that are coming out from XBVP. So as things go off the varietal protection list and become available, we've been working with that germplasm coming into tropical backgrounds. And there are a number of groups in the US who are working from that tropical background moving up. Certainly in terms of allelic diversity, when we identify something of value, yeah, it's free and open for people to start accessing that information. And I think this is where gene editing is really valuable. Crossing from tropical into temperate and temperate into tropical is not the easiest thing to do. You need to take that forward quite a few generations before we can conduct any reliable form of phenotyping. If we've got allelic targets that are editable, that gives us a much faster route to try to validate the potential value of some of these things. And depending on the target geographies, we can either move and continue through an editing route to deployment, or we can go back and do the harder breeding work, but with the security of knowing that that allelic variant will have value in an ultimate breeding product.
Mm-hmm. Okay, thank you. Question I have, or maybe like to hear both your and Kelly's perspective on, is AG2PI is now integrating crop and livestock. So the one CGIAR, Sarah, that you talked about also integrates, well, has centers, more crop-oriented and livestock-oriented centers. So how is that working in that arena, the integration of livestock and crop research and development?
In the CGM, it depends on the level and where we're at. I mean, I was strategically based earlier in my career. I was working for IITA, based in Africa, and I was actually positioned in ILRI. I was working in ILRI. And so there was a lot of opportunity there and real synergy between the livestock and the crop research in that more upstream space, looking at cost-effective genotyping technologies, etc. That was a real hotbed of synergy. We pulled a lot from the crop world from that livestock group there. So that was one big opportunity. Opportunities around data management, data analysis, etc. As we move more out then into targeting and production systems, there's a reawakening, I think, within some of the breeding communities from certainly the cereal perspective, that multi-use crops are very, very relevant. So many farmers, they get more income from selling the stover than they do from the grain. And so really making sure that we, in the product profiles that we're trying to design to, have captured fully the farmer's needs, whether it's from the livestock perspective or it's from the auto consumption or grain and produce sale perspective, is becoming increasingly important. So I think 1CG is a great opportunity to bring that conversation together a bit more.
Kelly, do you have any comments on this? Any activities integrating crops and livestock?
I don't really have a huge amount of comments on it. I think that considering the potential genotype by nutritional interaction, then there's a lot of opportunities. But then if you do select for specific nutritional substrates, then potentially if the substrates change, then the adaptation might be poorer on alternative substrates. But yeah, I think there is a huge opportunity to think about the G by E when it comes to the nutritional component.
Mm-hmm. Yeah, and and I think there's the systems approach of integrating livestock and crop production systems. I think there's both here and in the developing world, there's lots of opportunities. So. Okay, well, we're at the top of the hour. I want to thank both Sarah and Kelly very much for their presentations, and the audience for being here and for your participation. Couple of things as we close out. If you have ideas for future field days, please let us know. They are every third Wednesday of the month. Next month will be the 15th. There's also a crop modeling using the DSET software that workshop that will be held in June 23rd. And they fill up quickly. So if you're interested, register early. And then we still have some seed grant funding available for specifically for rolling out community event grants and working group grants. And you can get all the details on the website. So they are available on an ongoing basis until our grant closes or the AGDPI funding expires. So with that, thanks again, Kelly and Sarah. There is a Slack channel that has been opened to if you want to continue the conversation. And as you disconnect, you'll be directed to a survey, which we hope you will take some time to fill out. It's a very brief one. So. Appreciate it and hope to see you all next month. Thank you.